Here is a computer, so be sure to use your elbow to get past these bahunks here.
Please.
So there's a computer stashed in the corner there. So when you turn it on, you'll get your Windows login screen. And the login name that you're going to use is EMFAC, just like the EMFAC register. So it's EMFAC, all one word, all lowercase. And this is the digital camera. So very creative. Password is digital 4 camera, all one word, all lowercase. So just like reserved for me, digital 4 camera. When you log in, then it's going to give you this splash screen to tell you that you can't look at your Gmail and stuff here because it's not connected to the internet, so don't even think about it. There is a USB port on this, so you can plug that in. This icon over here that looks kind of like the wool symbol is called magnifier for magnification, and fire, F-I-E-R, not magnifier, F-I-E-R, it's F-I-R-E. So it's magnifier, F-I-R-E. So click on that. Let me go back a step. Okay. So start up that program before you do anything else is to start the program. So double click. And you'll notice it down in the corner here, it says camera off or not connected. So at that point you want to turn on the power supply, which is this Olympus up here, up on the top, not the lower one, which is that says Nikon. You want the one on top of it that says Olympus. And there's just a little toggle on and off, a 0I. So just push that. And you can see right now that the screen looks gray. So when I turn this on after the software is running, and only after the software is running, if things worked right, this should now go from gray where it says it's not connected to it is connected. So if it doesn't work the first time, if at first you don't succeed, turn it off again, turn it on again. And so now you would get a black screen if there was nothing there, or in this case, because I've got the light and everything on, you're all set. So this camera, this microscope is one of the old-fashioned ones. So you can see on either side it's got a little knob and a big knob, so it's got a fine focus and coarse focus. Your field aperture and your condenser aperture on the other microscope were these little rheostat things on the side. In this case, you have to physically turn them yourself. So this here is what turns the field aperture, and there you can see it getting bigger and smaller there. These two knobs here are the centering screws, as you can see on the screen here. This silver knob that sticks out is just a polarizing filter, so you're not going to use this for anything, so don't try to adjust this one. And hidden deep underneath here is a silver rod, and that's your condenser aperture. So everybody can see that. You're welcome to step forward here and give it a shove.
You mean this guy?
Yep. No, that's the centering screw. It's the one.
Oh, this guy? No, that's the. Is it the polarizing things? Oh no, this is.
Yeah, it's the only one that's going to swing like that. And so you can see on the screen as you're turning it that as you turn it at the beginning, it has no effect until you start getting it small enough where it starts to block the light in the image. That's about where you want to stop it. All right, so what's the first? Oh, and then let's see, what are the other things? This is for doing fluorescence. So in fluorescence, you're basically shining a bright light on your sample and then collecting the glow that comes off of it. And to do that, you have to use these things called filter cubes. So the light is that fluorescent light comes from the back here, comes forward, it passes through a filter, so to filter it so you get the appropriate wavelength that will make your particular dye in your sample glow. So that selected wavelength will then hit the mirror. It's a two-way mirror, so the short wavelengths of light get reflected down. Hits your sample absorbs it, they glow. That glow is then going to come up out of the sample into the lens and up to the mirror, where it then has to pass through the mirror so that it can then come to your eyes or come to the camera. And so there's a filter in the back so that the light coming, the multiple wavelengths of light coming from the light source get limited to just one or two or a narrow band. That narrow band of short wavelengths is reflected down to your sample. The longer wavelengths will then come up and pass through the mirror, pass through another filter that will give it the appropriate color like you see in the seminars, but which is all phony baloney anyway, because as you change your filters you'll get different colors. But they're close to it, so you might get a range from orange to red depending on what filter cube you're using. So you want to take those filter cubes out of the light path when you're doing bright field. You don't want your bright field to be red because you've got a red filter in the way for doing fluorescence. So you want to set it to the blank setting. So this is a carousel, so you can see here that there's no filter cube for this one. So you just need to swing it so that you're at the number 4 position. I'll have to put a number back on it so that you'll see it in your picture when you look through the eyepieces. You'll see a color there. So if you've got a color, it means you've got a filter cube in place. You need to rotate this and get it out. Come on, guys. Okay. So you don't need to take this cover off. That was merely, as they say, for demonstration purposes. All right. So what's the first step in doing color? First step is to get a focused image. Everything's based on having a focused image. Then the second step is to close down your field aperture. And now you can see when there's an on-off switch on the lower left side here. There's a rotary, there's a rheostat and an on-off switch behind it. So if you want to come and feel it, you can rotate it when you go to turn it on or turn it off. You want to push the rotary switch, the rotary dial, away from you, so that will turn down the current. Then you turn off the light. When you go to turn it on, you want to turn down the current if it's not already down, and then turn on the light, so you're not turning it on at full power to potentially blow out the filament. So you'll turn on, there's just this on-off switch with a rotary dial, and you can make the image brighter or darker. So you're going to adjust this so you can see the edges. These two knobs here will allow you to then center it, much like the other scope. And then you can raise and lower the condenser. So here I have the condenser far away and you're not seeing anything on the screen. And you can do this looking through the eyepieces. So as I'm bringing it up now, you're seeing here where now you're actually focusing on the dust in there. So this is the idea of what the Kohler illumination does, is it makes sure that you're looking at the illumination plane for the light rather than some other level in the light path. So you just bring that up, and it gets smaller and brighter, and then you just give it its nice sharp edges, expand it so you can't see it anymore in the image, and then adjust your condenser right here where you see it start to get darker. So it's wide open, I'm turning it, no effect, no effect, no effect, and now it's starting to darken. So somewhere right in here is where you'd want to leave it for your picture. All right. We have 3 lenses. Actually, we've got more than that. We've got a 10, a 20, a 40. Those are all dry lenses. And then we have a 100x. We've got 200x lenses. So you can use either of the 200x lenses. Remember that you're going to put a drop of oil on your slide and then put the oil lens into it. When you're done looking at it with oil on the slide, you have to clean the oil off so that you don't get it on the dry lenses, because once it's on the dry lenses it'll muck up your images. Alright, so now we want to take a picture of what we've got. So up here in the software along the top, this pair of binoculars is where you set it to find an exposure. So if I change the intensity here, or you go from a higher to a lower magnification, it's too bright. So then you're just going to click on the binocs, and it's going to go looking for the exposure and give you a fixed exposure. So now you want to take a picture of it. You have over here, you have a black and white called a monochrome snap, and next to it you have the color snap, RGB color snap, and next to that you have manual exposure color snap, which allows you to change the exposures for your red, your green, and your blue. And so you don't want to mess with those, just go to the automatic settings. So this camera is a black and white camera. You're all familiar with using black and white cameras because all of the cameras in your cell phones are black and white cameras. Well, how do you get color pictures from the black and white camera? On this camera, we take 3 exposures with a red, and a green, and a blue filter in it, and then you merge those pictures automatically. In your cell phones, they have micro-painted little lenses on your camera chip, so that instead of being 1,000 pixels square, it's 500 pixels binned, just like we were talking about binning for faster exposure on the other camera. Remember, instead of, we were taking 4 of these drop dots, we were binning them together so it's 4 times faster, because now you have 4 receptors collecting light as compared to having a single receptor for each of those 4 spots. So what they do is they bin, and then they paint two greens, 1 red and 1 blue, and then for that pixel then you're collecting a red, green, and blue image. But it comes at the cost. In a black and white camera you have 1,000 pixels. In a color camera you only have 500 because you bin them 2 by 2. So if you're doing really high resolution, high magnification work and you're looking for detail, the smallest object you can see is going to be dependent on whether you're taking it as a color picture or a black and white picture. And with your color cameras, you don't have the option because it's painted. You've got the colors painted on the chip already. So you can't say, well, this time I'm just going to do a black and white picture. All right, so here we can go ahead and take a color. We'll take a black and white snap. And there's our picture. And so this you can then go to File, Save As, and pull down into the D drive where it says Data, and then it says Users, and you can make yourself a folder in the Users folder for your data. Then your choice of saving. If you're saving a black and white, you want to save it as an 8-bit. Don't save it as a BMP, don't save it as a JPEG, any of these other formats. Don't save it as a Photoshop, because you won't be able to open it except if you have Photoshop. Alright, so we're going to save this black and white picture as an 8-bit, and then you would type in the file name and save it. Any questions on that? Pretty straightforward. Alright, so now we want to take a color picture. So we're not going to save this one. So color picture, just hit the color snap. And so now you can see the filter wheels going by. And it does it twice. The first time it sets exposures, and then the second time it's actually taking a picture. And so now there's your color picture. Now, if I change the intensity of the light, so now now this you want to save 8 bits for red, 8 bits for green, and 8 bits for blue. So you want to save it as a 24-bit image. If you screw up and save it as an 8-bit image, then you're going to end up with a black and white image. Alright, so, or you'll end up with an image that's all combobulated. So we'll get rid of this. So now I've made it much brighter. And as we talked about, at the lower light it looks yellowish, at the brighter light it looks gray. So search for an intensity with the binocs. And now when we go to take our picture,
so these are all filtered?
those are all, this is a filter wheel, the three position filter.
Oh.
See, now your color doesn't look quite right. So one way to correct for that is to say, okay, let me show you where white is. And so you come in here to camera, come down to white balance on left click. And so now you simply want to find an area where it should be white and left click. Now that will work if you're going to do it, but you'll have to do that for each picture. Now you can calibrate the camera so that you're going to say, okay, this is the intensity, 12.4. I'm always going to use that for all my pictures. So I'm not going to have to fix the color in each picture. So what we're going to do is we're going to, don't save this. You can see the little icon next to the binocs. It looks like a little yin-yang, black and white. So that's your white balance, auto white balance. And so when we press that, it says, give us a field that's all white. So you would move your slide to try and find an area. So this is the exposure you wanted because you already set that for your exposure in the black and white. So now you've got a white field, and then you say, okay, I've got a white field in front of the camera. All right, so now when we bring our sample back in and take that same color picture, and somebody asked in the other group, well, you know, why not just fix each picture as you go along? Well, this is easy to find a white area at this mag. When you get up to 40x, you may not have a white area in your picture to click on it to tell it to make this white. Or it's a small area and your right click white balance on the click is this big box. I don't know how many pixels it covers. So you may end up extending into some of your colored areas on your material. So here you can see now the color. We've got what appears to be a white background. So now the camera's set, so all the subsequent pictures, as long as you don't touch that intensity, should all give you the same color exposures.
Regardless of magnification?
Yeah, because it's the intensity of the light coming in. I'm sorry, it's the wavelengths of light coming in, not necessarily the brightest. So what you would do if, when you go to the next higher lens and it's dimmer, you would search for exposure, and it'll give you a longer exposure to compensate for the fact that you're collecting less light. But the wavelengths of light that you're collecting shouldn't be changing. So the color shouldn't be changing just because you change the mag. All right, so let's see. So we don't see it so much here. If, just like on the camera, if you had something here and like this smudge here, if when you moved your sample it didn't move, then you would know that it's in the light system somewhere. And so you would take your picture like this, with this, that's going to show up in all your pictures. So when you have sample covering it, it's still there, but it's harder to see because you've got sample in the way. But if you end up with a clear area and that's there, that will come through. So you can take this picture and that becomes your background, and you would save it as a background. Alternatively, you could, if you didn't have a space where you had dirt and precipitate from your staining everywhere on the slide, then just make up a fresh slide that just has your clean slide with a drop of oil plus a cover slip on it, and use that for your blank field. And then anything that's in the light path will show up in that picture. So now you have your background picture and you have all of your sample pictures. And then you would go into like image J, and it's called, I think it's called background correct, or flat, it's called flat field correction. And so what the flat field correction is going to do is it's going to take this picture and subtract it from all of your sample images. And so that will remove that smudge from all of your pictures. If you have anything else in there, it's also going to be subtracted. And so you may end up with the inverse artifacts of having done the subtraction. You've now created an artifact in your picture by pulling out that data, because now it's changing the brightness of, say, a spot in the middle of your sample. So you have to be careful when you're doing this background correction stuff. Okay? So it wouldn't hurt when you sit down to take pictures to always take a blank field. If you don't do anything with it, it doesn't occupy much space, it didn't take you much time, and you have it. And then if you look at the lab report instructions, and it says make sure you include this, or you want to show off that you're hot stuff and fix all your pictures so they don't have that piece of junk in there that you didn't figure out where it was, that would be the way to get around it. Okay?
Cool.
So I'll post how to do that in ImageJ, because that runs on everybody's computer and it's easy to get.
And this you want us with pre-made slides or the ones that we cut ourselves?
Oh, at this point, everyone supposedly has got thick slices. So yes, use your own stuff. Well, use your own slices. And then, as I say, you can just the first report's just going to be black and white, and then the second report will be color, but you can use the same set of slices, so you don't have to do it all over again.
Cool.
So it won't take up too much of your time. Okay?
Fantastic.
But this is a problem that I know. I recently had someone come in and say they were having problems using a color camera. This is probably the most common thing because, you know, people don't teach people how to use this stuff. They just turn this knob, turn that knob, take your picture, and you're done. And they don't appreciate having these differences in setting your light intensities. So sometimes people don't know that and turn down the light intensity because it's too bright for them, because they're not dealing with the samples everybody else is used to. They're slices and you're using individual cells. So that you have to be careful with that, because it has an effect on your images. And it's a pain to try to go back and fix it later.
Dig it.
Okay. That's it.
Awesome.